for giving me this opportunity to present my research. And this is very much work in progress, so I very much appreciate any feedback that you guys might have. So to start off, the genome contains many different regulatory elements. For instance, there are active genes, there are repressed genes, and there are like promoters, enhancers, and insulators which regulate the activity of these genes. And one feature of these regulatory elements is that they are associated with different combinations of histone marks. So the other feature that these elements are associated with is accessible chromatin. Because usually these regulatory elements are functional only when transcription factors are binding to them. So the chromatin has to be accessible in order for these transcription factors to bind. So a common approach to identify these regulatory elements is to use the combinations of different histone marks. So here, I'm showing you a region which contains an active gene and a repressed gene. So the active gene is on the right side of the figure, and the repressed gene is on the left side of the figure. And if we look at the combination of histone marks actually near the promoter of the active gene, we see that it has high levels of the trimetal mark at lysine 4 and the acetyl mark at lysine 27. And this combination of histone marks is associated with active promoters. So to the left of that active promoter, we see a combination of monomethyl mark at lysine 4 and acetyl mark at lysine 27, which is associated with active enhancers. So by looking at the various combinations of histone mark levels across this region, we can actually segment the region into different regulatory elements. So to actually perform this segmentation process given the histone mark data, a common approach is to use ChromaHMM, which is a hidden Markov model that basically clusters these signals into distinct chromatin states. So here I'm showing you on the left the emissions matrix of the hidden Markov model, but every row in the emissions matrix represents a distinct combination of chromatin marks, and every column represents the signal level of a particular chromatin mark. So I broadly classified this relatively large number of states into a small number of 8 states, which includes like a promoter state, which is associated with, again, the trimetal mark at lysine 4 and the acetyl mark at lysine 27 mainly, and transcribed states, enhancer states, CTCF insulator states, heterochromatin, poised states, repressed states, and quiescent states. So while this approach has been very successful in annotating many genomes, one key limitation is that it requires, as you see, multiple histone ChIP-seq data sets. And each of these histone ChIP-seq data sets requires millions of cells, and it is a costly process. So how can we actually do this if we're looking at, for instance, a small cell sample like primary tissue, or like primary tissue, or like rare cell populations? So one thing that we notice is that a lot of these regulatory elements are actually associated with open chromatin, as reflected by the high levels of DNA signal. And we could potentially just do DNase, but DNase also requires millions of cells, so it's not really feasible for these situations. So our collaborators in the Greenleaf lab have developed a new assay that is able to perform a genome-wide chromatin accessibility from low input material. And the way that this actually works is that there's transposons loaded with sequencing adapters, which then cut the genome at accessible chromatin sites and inserting these sequencing adapters. And at the same time, basically fragmenting the genome into different size fragments. And we then sequence these fragments using parent sequencing to obtain a genome-wide map of accessible chromatin. So one important point here is that these peaks that you can get from the ATAC-seq signal actually would correspond to regulatory elements, just like DNase peaks correspond to regulatory elements. But this picture actually doesn't reveal the full power, so to speak, of the ATAC-seq assay, because it only shows you the information about the cut size, the accessibility. But because we do parallel sequencing, we actually, in fact, get more information about the fragment lengths of these different accessible fragments. So, for instance, if we looked at, the global fragment length distribution that we obtain this is obtained in a human lymphoblastoid cell line, we can see that there are unique peaks peaks at different fragment lengths, which are actually associated with the different aspects of nucleosome architecture. So there is a noticeable peak at about 50 to 100 base pairs, which are associated with the nucleosomal free fragments. And the next peak further on at about 200 base pairs is associated with fragments that are protected by a single nucleosome. And like the peaks at even larger fragment lengths are associated with basically DNA that is wrapped around multiple nucleosomes or like heterochromatic regions. So what I'm showing you here is actually just a genome-wide plot of the distribution. And the question is, what happens when we actually look at this distribution when we add different regulatory elements? So here, I'm actually showing you a histogram. Basically, I'm flattening the distribution down to one dimension. And you see the red regions correspond to higher density, and the blue regions correspond to lower density. So here we see that across all promoter states in the genome, there is an enrichment for short fragments, again, for 50 to 100 base pairs, and also at like various multiples of the nucleosomal size, like 200, 400, 600. So I mean, this is consistent with what we know about promoters, that they have to be accessible for transcription factors to bind. So looking at the other states, we see that the different chromatin states are actually associated with different, maybe subtly different fragment length distributions. And suggest that the architecture at these different regulatory elements reflects their state. Again, though, this is actually an aggregate plot, which is aggregated over multiple sites of the chromatin of the regulatory element of the particular state. And I mean, we know that there's various problems with these aggregate plots, that sometimes due to heterogeneity, these plots may not be actually representative of individual sites, the distribution at individual sites. So the question here is the fragment length distribution actually predictive at individual sites? So in addition, another thing that we're not actually capturing using these fragment length distributions is the positional information that's provided by the assay. So for instance, by anchoring on, for instance, ATAC-seq peaks that are in the CTCF state, we can actually plot a two-dimensional histogram of the fragments, of the density of fragments, where in this plot, the x-axis represents the relative position to the peak summit, which is in the middle, and the y-axis represents the fragment length. So for instance, this enrichment or high density region near the peak summit of fragments between 50 and 100 base pairs, it corresponds to the accessible binding site where CTCF is likely to be binding. And the enrichment of longer fragments of 150 to 300 base pairs corresponds to these position nucleosomes that CTCF is often associated with. So again, this is still an aggregate plot. And let's actually see what the data looks like at the single site. And we see that it's way noisier. And it's very sparse. So we are actually going to try to identify the chromatin states from these two-dimensional fragment length distributions at ATAC-seq peaks. And so from a biological perspective, if we are able to do this, it would show that there is a clear relationship between the chromatin architecture at ATAC-seq peaks and the actual chromatin state. So we have chosen to formulate this prediction as a supervised multiclass image classification problem, where given a set of fragment length distributions at the ATAC-seq peaks, two kilobases around the ATAC-seq peaks, we will try to determine which of these 8 chromatin states it actually lies in. So at this point, I mean, many of you are familiar with machine learning. You could use your favorite machine learning algorithm to solve this problem, but we decided that since this is actually like an image classification task, we could leverage recent advances in image classification technology to solve this task. And one model which has become pretty dominant in image classification are deep neural networks. So, for instance, as a toy example, suppose one typical image classification task is face recognition, and suppose we were trying to build a classifier to recognize images of my advisor Anshul against Manolis, who is one of the organizers of this conference. So we given a set of images of Anshul and Manolis, we can train a deep neural network that automatically identifies the relevant features that are characteristic of these two people, and then use that to classify whether the image is one of Anshul or Manolis. So again, I emphasize like these methods are hugely advantageous in that we feed in the raw image pixel values and we obtain a classification, and you don't really have to build the features by hand. So on the right, I'm just showing you what other people have, the kinds of features that other people have managed to learn from a deep neural net when they applied it to faces. And like in the first layer, you see like edge detection features, which are then combined to give you like smaller pieces of the face. And then on a higher level, you get more larger sections of the face. So for instance, if we had a picture of Manolis, we can, like the relevant features that correspond to him will be activated in the network, and we get a correct classification. And if we put in a picture of Anshul, a different set of features gets activated, and we get the correct classification of Anshul. Yeah. So now we're going to apply the same technology to classify these fragment length distributions. So assuming we put in the fragment length distribution in the bottom, we hope that we will be able to learn features such as nucleosomes or like regions of open chromatin, and then segments of open chromatin, and then nucleosome arrays, which, for instance, in the case of CTCF, would be the distinguishing feature that allows us to classify the fragment length distribution as belonging to a CTCF state. So how does our method do on this task? So we took ATAC-seq data from the lymphoblastoid cell line GM12878 in humans, and we did a multi-class prediction using this model. So again, we're trying to predict one of the 8 classes: promoters, enhancers, insulators, etc. And we see that we outperform greatly several baselines. So the first baseline which we outperform is the majority class baseline, which is the simplest thing. You just predict everything being the most common class, which in this case is enhancers. And then a stronger baseline would be to use existing annotations of transcription start sites to classify these things as being like whether they are promoters or not. So that does much better. That does better than just predicting everything as an enhancer, but it still does much worse than our method. So this suggests that chromatin architecture is reasonably predictive of chromatin state. So one limitation that we notice is that these labels are actually not they are not ground truth. They are actually noisy, and we can actually use data from replicate histone chip-seq experiments to estimate the noise in these labels. And when we did this, we noticed that approximately 12% of the labels actually switch across replicates. And when we evaluate our method only on the consistent labels, we find that we get a boost in our accuracy. So actually, we are doing pretty well considering that this consistency between replicates gives an upper bound of the accuracy. And finally, we decided to also look at how we could do on a specific state. So we chose the CTCF state because we actually have ground truth for this state in the form of chip-seq peaks from CTCF chip-seq experiments. And so here we are trying to determine if there is actually a CTCF chip-seq peak within one kilobase of the ATAC-seq peak. And we see that our method performs much better than stuff based on motifs and DNAs or just predicting everything to be CTCF bound. And the reason this is so is because many of these CTCF chip-seq peaks do not contain the canonical motif. So you wouldn't find it if you had to rely on the presence of a motif in the first place. So in conclusion, we have quantified the relationship between chromatin architecture and chromatin state by producing a predictive model that actually is able to determine chromatin state using a single assay with reasonable accuracy. And in addition, our model also recovers CTCF binding sites using only the chromatin architecture signature, which is that it basically phases nucleosomes around it. And so with that, I will go to my acknowledgements, and I would like to thank my advisor and Johnny Israeli, who is like the rotation student who worked on this with me, and many members of the Greenleaf Lab for providing data and many valuable insights on the ATAC-seq assay itself. Thank you.
So I think we have time for one question, and if the next speaker takes place, yeah.
So it seemed like you were training and evaluating on the same cell type. It seems like some of the real power of your method is if you could train on one cell type and then apply it to a different one. Have you evaluated if you lose performance?
We are collecting the data for that because we find that you actually need pretty deep data for doing this. Yeah. So our collaborators are currently generating the data on another cell type, and we're going to try it. Yeah.